Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Kasper Roet, CEO and co-founder of QurAlis. Kasper is also a 2022 Tamir Foundation Fellow. Today, we will be discussing how drug development is changing, giving hope for transformational treatments for patients with ALS and other neurological diseases. Welcome, Kasper.
Thank you.
So let's just get started. Your early research really focused on spinal cord injury before you shifted to ALS. What was that influence, and why did ALS become kind of your focal point for research and what you do today?
Well, that's a great question. I actually wanted to develop therapies for patients for a very long time. And spinal cord injury, I did my master's internship in that space, and I thought, this is a tractable problem. That's how I looked at it. There's an injury to the spinal cord. We know the problem is in the end that the nerves are being cut and that we have to reestablish these connections that are lost. And obviously, it's a huge liability for patients that are paralyzed. And I'm sure you know people like that. And so I thought I can really understand this and I can make a difference. And so I did a PhD in gene therapy for spinal cord repair. This was in the early 2000s that I was doing my research there in the Netherlands Institute for Neuroscience. Did also a master's internship at UCSD. But because the lab where I was working was also focused on ALS, because in the end, it's also neuromuscular disease. The cause is different. In ALS, you basically have a disease that is taking away the connection between the neurons and the muscles. In a spinal cord injury, it is an injury itself. But for ALS, I couldn't understand how it worked. And so there was only one gene that was at that moment known to cause ALS, SOD1. And it only explained ALS for 2% of the patient population. And we didn't know even how it actually was causing disease. And so for someone who is like me, who wants to be able to solve a disease so that we can do something meaningful for patients, that didn't feel like a disease where I could actually make a real difference. And so I was working with the Dutch Brain Bank. I was working with patients and their families who wanted to donate their tissues to help scientists understand disease and potentially create solutions for patients. And so I was very closely involved with diseases, many diseases: Alzheimer's disease, ALS, Parkinson's disease, frontotemporal dementia disease, multiple other diseases. But I didn't know how to make a drug. And so then I realized, okay, after I finished my PhD, I need to understand actually how you get something from a genetic insights to patients. I started working at Johnson & Johnson, who had a vaccine development institute and center in the Netherlands in Leiden. And I worked there on an inactivated polio vaccine. And so if you know polio, then you know this is also a paralysis disorder. And polio is actually a virus that in a small proportion of patients gets into the motor neurons and creates havoc there and actually leads to motor neuron death and paralysis. So it kind of stayed within my realm of experience. But what I learned at J&J, first how to develop assays, but then also how to actually take something from a scientific discovery in the research side and, through analytical development, upstream and downstream processing, CMC and manufacturing, formulation, fill and finish, quality, clinical assays, and then bring it to the clinic. And so that gave me an understanding of how you actually develop a therapeutic. And then while I was there, C9orf72 was discovered. And a big ALS gene, basically explaining about 8 to 10% of the patient population, both sporadic and genetic. And then rapidly other genes were discovered as well. Suddenly, I felt ALS is no longer a disease we don't understand. We have 40 different genes that can cause disease. And if you map them out into how a cell works, we can see that specific pathways start to emerge. Very clearly, an autophagy pathway started to emerge. Cytoskeletal pathways started to emerge. Cell stress pathways started to emerge. And I thought, now I probably can actually do something for ALS because this is now a disease where we have more genetic understanding of what is going wrong. And I realized with my background, very highly specialized in the motor system and the neuromuscular system, I have a really good opportunity to do something meaningful. So that is really how I ended up focusing on ALS, really on the basis of that genetics.
And don't you have somewhere in your educational background something around like networks and information management that has helped you kind of mapped? Do I remember this correctly? There's something kind of interesting in your background there that has kind of helped you to layer this understanding.
Yeah, so you have a good memory, and Deborah. I thought I wouldn't go into that level of detail, but since you ask, so I'm a big data person. I love a lot of data, and now fortunately that is a very good thing. And so I started actually my university path with artificial intelligence, but I realized my expertise is even stronger, or at least is stronger on the biology side, specifically the medical biology. But I've always been very close to data and biology. And so a product of the Human Genome Project, which was amazing, right? Like deciphering the human DNA. And so we figured within no time we'll understand all diseases and have therapies for everything. Well, that didn't pan out. But what happened is that we did suddenly understand DNA, and then we started to understand RNA. And RNA was giving us, it's the transcript between DNA and protein is RNA. And so what RNA was giving us is kind of a blueprint of everything that is happening at the cell at a certain moment. And so we had RNA probes, microRNAs and RNA-seqs from all the different genes. And so what I was doing, and was also part of my PhD, was to create a meta-analysis of all of the published RNA data sets, both RNA-seq and microRNA. And so what you get is a quantification of which RNA molecules there are in a cell, and you can use that to compare disease versus healthy. And so in my case, in my PhD, I did that on the regenerative system of the olfactory bulb. But when I was at J&J, I started applying that to ALS. And so I looked at all of the post-mortem tissue, so patients that had donated tissue to brain banks, and all of the published data sets. And it gave me basically hundreds of thousands of RNA molecules and how much was expressed in patients with ALS and how much was expressed in healthy individuals. And I started to combine these all to find patterns that are conserved between all of these different studies, between a lot of different patients, looking at whole tissue, but also at single neurons that were taken out of the tissue with a laser and single cell RNA quantification, basically. And out of that came a list of genes that I thought explain disease in ALS patients when I compare that to healthy volunteers. And so that is my passion. And so I had this list, and I thought, now I have potentially targets to develop therapies for patients. But then I thought I need a model to see if these targets are real, if this is actually causing disease, and also to test potential therapeutics. And so I decided to build the same type of database with animal tissue. So there were mouse and rat data mostly, with SOD1 mutations mostly, but also some other mutations. And I thought I should look at if there's the same patterns in animals as I see in patients. Because if I can find the same patterns, then it's something I can test. And then if I can find something that works, that it kind of like corrects the disease in an animal, I can bring that also to patients and hopefully correct the disease also in a patient. That turned out not to work at all. There was zero overlap between the list that I found from patients with ALS and the list that I found from animals that were used as ALS models. And so that was very disappointing for me because I was like frustrated because I thought I have potential genes that we can target to make therapeutics, but I don't know how to test them. And you cannot just bring something to a patient. And this is when I discovered Kevin Eggen's work at Harvard. He was a professor there, and he was working together with a doctor at Massachusetts General Hospital, James Berry, and another professor in Japan, Yamanaka, had recently discovered how to make stem cells from skin cells. And so Kevin was a developmental biologist, and he took the skin cells from ALS patients in collaboration with James Berry, turned those into induced pluripotent stem cells, and then applied his knowledge to turn those into motor neurons from patients again. And so, for the first time in the world, we could actually study the cells that are dying in a patient's spinal cord in a dish by creating them, basically recreating them from their skin cells. And I thought, okay, if we can use someone's own cells and to make the neurons that are sick and in the end dying in their spinal cord, that should be a translational model. If we can find things that are going wrong in those cells, then those should be representing what is going wrong in the patients. And if we can potentially treat it with a potential therapeutic, then we should be able to have something that actually is meaningful also for patients. And so that made me quit my job at J&J, and I left to Boston because I wanted to learn that technology, because I thought with genetic understanding and these patient-derived stem cell models, these neurons from patients, we should be able to develop meaningful therapeutics for patients. And so that, and so my data passion kind of brought me to that conclusion and brought me actually to move from the Netherlands to Boston and work at the Boston Children's Hospital and the Harvard Stem Cell Institute for a number of years.
Yeah, I've talked to other researchers and entrepreneurs about these pluripotent stem cells because you remember and I remember several decades ago, the discussion around stem cell brought a lot of emotion and a lot of concern, but the technology has gone so much further. So I'm glad you brought it up. And when you took the stem cells from each one of the ALS patients, that kind of leads us to really precision. Was there any kind of differences that you were seeing in some randomness there, or was there more consistency across, the therapeutics that might apply?
Yeah, that's a great point. It's one of the challenges working with stem cells that there's also a lot of variability. So basically, if you take skin cells from a patient and you create stem cells, all of them look a little different. And so there are differences even from one patient. And so you create neurons, then you need a lot of quality checks along the way of the whole process. And it's a process that takes multiple months until you actually have from a skin cell to an induced pluripotent stem cell, and then the differentiation to a neuron. And so we've learned to really focus a lot on the quality during this process. And so there's a lot written down on that. But in the end, what is also really important that you really have expert people that understand also what the difference is between a real stem cell and a stem cell that is partially locked already, for instance, in differentiation, because then you can get a different outcome. And so this has been a challenge for the field, and it's been a criticism for the field because that variability can also lead you on the wrong path. And so, especially in the beginning, there were a lot of skeptics, even at Harvard, that thought you could better use a kidney cell from someone to study a neurological disease like ALS than to create a stem cell from a skin cell and turn that skin cell into a motor neuron. Now, I didn't believe that. And I was actually kind of surprised when I got there that there was so much controversy about it. But I think the field has matured a lot, as you said, and we have gotten much better in building in quality checks. But it's still not trivial. And if you, for instance, we're very good now at creating monolayers of cells, so basically we can create neurons that are on one plane. But if we create organoids, which is a very interesting technology as well, it's even more complicated to really make sure that you can get. So an organoid is basically the beginning of an embryon. And so there's a lot of developmental cues that go into that. And having reproducible organoids is really that have the same shape, that have the same division of different cell types is a very difficult thing to do. And so we're still struggling with that in some areas, but we've also kind of mastered it in other areas. If you look at our company, QurAlis, we've solved that by bringing really the best people we could find from different stem cell labs together. And, they are better at it than I ever was, honestly. And so now I think we have the skills to do it, but we also know where our limitations are. And so if you do it the right way and you do it with sufficient different cell lines and you get consistent results out of that, then it is real. And what we always do with QurAlis, and we did that also as part of the Harvard Med School, we always looked at patients and made sure that what we are seeing is something that we could find back in patients. That kind of makes it believable for me. If you just see something in a stem cell model, in the end, it's just a model. And honestly, the stem cells are kind of embryonic. They represent an early stage of development, not when the average age of onset of ALS is 55. We don't have 55-year-old neurons. And so there's a limitation also in what we can get out of that. But if we have a really good quality procedure, we get a reproducible result with many different cell types from different patients, and we see that also back in patients' tissue, then we think we have something real. And now others in the field start to replicate it as well. That really gives us the confidence that this is really real. And so, that is something that we're hyper focused on. Academic groups are really focused on making new discoveries and publishing groundbreaking new science. Companies want to make sure that what they're doing is real. And so that's really core of that quality trajectory is core of our programs.
So tell us about your company. Tell us about how did QurAlis come about? How long have you been around? Just that journey to being a research entrepreneur.
Yeah, so QurAlis is now 8 years old. We founded the company in December 2016. And so that was basically 3 years after I arrived in the United States and working with Kevin and Clifford. QurAlis is co-founded by myself and Clifford Wolf and Kevin Eggen, two professors at Harvard that were collaborating on these stem cell technologies, together with another company which was helping us get started, QState, which is now called Quiver. And so the company was created, and you can see it in the name, to cure ALS, QurAlis, cure ALS. But we were creative with the name also, and I think you alluded to it earlier. Alles also means everything in Dutch, and I'm Dutch, and my co-founder is South African, and so he understands that as well. So we always set up the company with a little bit of a broader purpose. Obviously, our first goal is to put an end to ALS, but I'm a strong believer in we have to develop therapies for all diseases. Honestly, we should, with our knowledge and technologies, we should be able to fix diseases. But my journey, I was mentored by an experienced CEO and venture capitalist, Jonathan Fleming. He really taught me how to set up a company and how to mature the company and which things are important while you're doing that, culture being important for that. And the start of the company was based on the first big discoveries with the stem cell technology. And so the first big discovery using motor neurons from a patient who had an SOD1 mutation was that a potassium channel in those motor neurons wasn't functioning properly. It had a loss of function. And so this was discovered with this collaboration between Clifford Wolf and Kevin Eggen and a couple of very talented postdocs in their labs, Brian Wenger and Evangelos Kiskinis, who both now are independent PIs, principal investigators. But they together discovered that there's a loss of the potassium channel current in these motor neurons, and that leads to neuronal death. And so that this was a very big discovery in the field. They also discovered that if you bring the channel function, so the channel name is Kv7.23, if you bring the channel function back to normal, you can protect these motor neurons for their survival deficit. And so basically bring survival back to a normal level. And so before we started QurAlis, academically led, together with GlaxoSmithKline and the Harvard Stem Cell Institute and the ALS Association, and I think UMass, Mass General was part of that, a trial was started because there was an approved compound on that potassium channel. And the trial worked really well. And some biomarkers that predict survival actually normalized, but the compound was not tolerated very well. And so that was actually when we decided to start QurAlis, to come with a better molecule which had the same positive effects on target properties, but had a better tolerability profile and not have the off-target liabilities. And in the end, QurAlis was started on that first big discovery on the stem cell field, and that is now a clinical program that is running. It's called our QRL 101 program. It's a small molecule to bring the potassium channel function back to normal. And again, in our models and in that first trial, that seemed to have a protective effect on motor neurons. While we were progressing that program within our company, if you think back about my big data analysis and my list, on the top of my list, I had another protein or RNA molecule called STAFMIN2, a microtubule stabilizer protein. Now, I didn't know what to do with that. And so, again, I moved to Harvard because I wanted to learn the stem cell technology. But Kevin actually had in his lab some scientists working on that particular protein or RNA molecule, STAFMIN2. And so while we started QurAlis, the science around that target started to mature. And it was Louise Williams, together with Joe Klim, and in his lab, that discovered it is regulated by TDP43, and that the protein is lost in most ALS patients because of a missplicing in the pre-mRNA, leading to a loss of RNA and protein. And that, we thought, is an amazing discovery as well. And we started another clinical program, we started a program on it, which is now in the clinic. We're dosing patients in 6 different countries. That we decided not to do with a small molecule, but with an antisense oligonucleotide to correct the missplicing of STAFMIN2, which then brings the RNA and the protein levels back to normal, which we hope will have a really profound effect on patients, because this particular protein is essential for the innervation of your neuron on your muscle. And that innervation is kind of a hallmark of disease. You lose the fibers going into the muscle, leads to a loss of muscle control, atrophy, paralysis. And in ALS patients, once it reaches the lungs, they pass away.
Do we know why there's a loss of function that occurs with this potassium channel, or do we just observe the action and then kind of react to that, or try to find things to reverse it, stop it, or reverse it? Do we know why?
So both of these, potassium channel Kv7.2 and STAFMIN2, are linked to TDP43 function. And so TDP43, the discovery of that pathology in ALS, has also been a big breakthrough. About 97% of patients have TDP43 pathology. And it turns out when patients have that, TDP43 is a DNA and RNA binding molecule. And it actually binds Kv7.2 pre-mRNA, and it binds STAFMIN2 pre-mRNA as well. And so when TDP43 moves out of the nucleus, it leads to a dysregulation of these RNA molecules and causing missplicing. So in the Kv7.2, a skiptic exon is introduced in the RNA, and the skiptic exon actually leads to a non-functional protein. So the ion channel becomes non-functional. That is degraded, and it leads in the end to about a 30% loss of channel function. And so that explains why the channel is decreased, and that decrease then leads to motor neuron death. Now, to make it more complicated on that potassium channel, hyperexcitability, which is caused by the loss of that function of that channel, also leads to TDP43 pathology. It leads to TDP43 moving out of the nucleus. So what you get is kind of a negative feed-forward loop of neurons that are hyperexcitable, that are then having TDP-43 dysfunction. It's kind of that's a stress response. Then the TDP-43 dysfunction itself leads to a loss of Kv7.2 through the skipped axon being included, leading to a non-functional protein. So we think we can break that loop with a Kv7 opener and bring the brake back basically on the excitability. Staffin 2 is a cryptic axon that is normally masked by TDP-43. So TDP-43 sits on the pre-mRNA and masks the cryptic axon so that when transcription takes place, that the cryptic axon is not exposed and it's kind of skipped. If TDP-43 moves away, again, because of a stress response, then that cryptic axon is exposed. It has a transcriptional stop codon in it, and very rapidly transcription stops. RNA is very truncated. There may be translation. We've never been able to pick up a short form of the full protein, but full-length protein is definitely disappearing. And so, again, that protein is essential for the innervation of your muscles. And so that has a very negative effect on the motor neuromuscular system, basically. And so, so TDP-43 is really an important regulator there.
You go through this stuff and I just, not being a researcher, not understanding, not coming from that background, it's so complex. It's just amazing that we've gotten this far in the understanding. And the other thing, as you were talking, is, your passion is to cure diseases. But then making that leap from being a researcher to an entrepreneur running a company, these things are very different. They're very different languages. They're very different, tribes, if you would. Talk about that transition and maybe even expand a little bit and tell about, some of the mentorship maybe that you got from the Tamir Foundation through the fellowship.
Yeah, I'd be happy to. So first, I think I should explain, I had a company before, after I did my bachelor's. It was an IT company. So I had some entrepreneurial experience there. And honestly, before that time, I never thought I would start a company. Like my parents, my father was an engineer, my mother an arts teacher. And having my own company was never on my radar, basically. But I kind of rolled into it because I was good with computers. And the rise of the internet in the 90s, and there were a lot of people that didn't understand it, and I did, and some friends did, and then suddenly we were like asked to help everyone, and we started a company out of it. But I also realized that's not what I really want to do. I was really, I'm a biologist at heart, and so, but it made me realize that you can create a company, and then I realized also that I want to do something for diseases. I experienced, like many people, a lot of diseases in my life with family members, and I thought we should be able to do something about that, and it kind of started my path. So from that moment, the PhD I did was a collaboration between companies and academic centers. So I actually worked together with AMT, Amsterdam Molecular Therapeutics, as part of my PhD project. I said it was a gene therapy project. AMT was a gene therapy company. It later became known as Unicure. Unicure is also present here in Boston. And they actually had the first approved gene therapy in the Western world on the market, Glybera. And I was there when they were actually having that success. We were working with them. And then also worked with Galapagos, which was another company developing gene therapies, but then for research, adenoviral therapies. And they actually kind of linked together with J&J, where I later ended up. But I was interested in that because I wanted to, again, be both scientist and business was always important for me. And so that's why I worked at J&J. And I worked as a fellow at the Harvard Med School, not in essence to do the research, but because I wanted to learn the stem cell technology. I'm just saying that because I've never been the traditional pure scientist. I've always been both, basically. But they call me a scientist entrepreneur. And so you said maybe it's very different to run your own company to being a scientist. But when the main job of the CEO pretty much is to raise money so that you can actually progress the programs. the investors that we are talking to, the analysts that we are talking to, they usually have PhDs and they're scientists. And so their knowledge and understanding is very, very deep. And so I always find it amazing how people that are not scientists can do this job, because I get so many complicated questions all the time. But what the Tamir Fellowship did, that what the Tamir Foundation did, is two things, which both are incredible. It's a network of really experienced, very successful CEOs and some other pharma leaders. And so and they provide access to that through mentorship. And so I said, when I started QurAlis, I had a mentorship from Jonathan Fleming, who was a very experienced company creator. But these people, like John Mariganori, who was really behind the success of Alnylam, just to give you an example, these people suddenly were accessible to me. And so I could ask really specific questions at specific growth stages about how to work with a team, how to work with a board, or work with investors, how to think about strategy. That's been incredibly valuable. And at the same time, you get a peer network from other people that have been awarded the Tamir Fellowship. And in my case, also a Transatlantic Connections Fellowship. Other CEOs that are more at my stage, so earlier stage of biotech, and you can share your experiences and often your frustrations also, and the challenges that you've encountered. And just to be able to, because in the end, as the CEO, you're at the top of the company. And so there's not many people where you can share these things with, but you can with peers in your field. And so both mentorship and a network of peers together really has been very powerful. And I think that I'm very grateful that I was able to be a part of that, or am able to be part of that. The other thing that the Tamir Foundation brings, and this is really why it was created, Henry Tamir, who founded Genzyme and kind of is the father of the rare disease space, because before Genzyme, there wasn't actually an active, well, there weren't a lot of programs or companies active in that space. He really kind of pioneered that. But he always put patients first. And so that was really what he was known for, that you have a very patient-centric focus in your company. And the second thing he did was mentorship. He always made time, apparently. I've never met him. I wanted to meet him, but he passed away right before I was supposed to meet him. The other thing that he did is making sure that you understand that as a leader in the biotech community, you have a responsibility to also help and develop the biotech community. And from his perspective, that meant mentoring motivated and promising individuals. And what I've heard, he had a really broad, open door for people. I met an investor actually last week when I was in the Netherlands who had exactly this experience with Henry Tamir. And he thought he was just surprised that the great Henry Tamir just wanted to see him and help him, even though he didn't know him at all. And so these two things, learning what it means to really be patient-centric and have this mission-driven culture, and passing that on the next generation. That's what the Tamir Foundation is really about. And I think that's fantastic. I mean, it really helps you focus your decisions on some kind of moral compass.
So you're at that 7-year, 8-year mark of a startup, which are tricky, right? A lot of them don't make it past the 7 years. What are the challenges that you find yourself right now? I mean, you mentioned fundraising. I'm sure that's a big one. But what are some of the other challenges that you find, that you're dealing with right now?
I've never looked at it like that, but now that you mentioned that, it's very similar to relationships, isn't it?
Absolutely.
When I started QurAlis, people said that most companies don't survive 1 year. And, when I think about relationships, the 3 years is one of these marks as well. I never saw it as a 7-year mark, but you're completely right. That is kind of a human transition point. The challenges are always the same up to now. It's always, getting to the next inflection point, meeting a new critical milestone in development. And just to give you a little bit more, I guess, make it more concrete. We started with pretty much nothing, the identification of targets. then the next inflection point was to get to lead molecules and really validate the target, basically. And so that was all part of the seed stage. Once we reached development candidate stage, so you have a molecule that you want to bring towards the clinic, that's another big milestone in a program. Once you're at that milestone, then the next milestone is to get to the clinic to have a filing of an IND or a CTA. Once you have done that, the next milestone is safety. Once you've done safety, then often the next milestone is target engagement. And once you have target engagement, the next milestone is efficacy. If you have efficacy, you need to repeat it. And then usually you have an opportunity to register the drug. And so there's always the next milestone to work towards. And if you have different programs at different stages, then, these programs are going to be at different milestones. Now, in a biotech company, your most advanced programs are the ones that dictate your value. And so those are the most important, because the further you get, the more expensive it gets, but also the probability of success gets larger. And so that there's a value associated with that. So where we are now with QurAlis, we are basically with our StaffMin2 program, we're dosing patients in 6 different countries. And we're going to look at efficacy biomarkers in parallel to our target engagement analysis. We already have some really good sense of tolerability. We have a really good sense on PKs or concentrations in the cerebral spinal fluid. Now we need to find out if the drug is actually doing what we want it to do, and does it have an impact on patients. So that's a very big milestone for us. So that's where we are now. And with the KV7 program, we're a little bit earlier than that, but it's a small molecule program. So there we have basically done the tolerability, and now we're at the target engagement phase. And then the next phase will be to see if it is really efficacious as well. But you talked about challenges, and I said it before, the CEO's main responsibility is to set strategy and to make sure that we are sufficiently funded to execute on that strategy. That has always been the same. So it started when I started the company. It's still the same now. I think it will be the same for a long time after this. But I'm actually really excited of where we are as a company. I'm really excited where we are as a community also. And I'm excited because a couple of things have started to click together in a very interesting way. So again, we started QurAlis to develop therapies for ALS. We have 3 programs that are actually targeting 3 genetic targets in ALS. I didn't talk about the 3rd program, the UNC13, which is a synaptic vesicle release, which we partnered with Eli Lilly. But with that, we are the leading company at this moment for therapy development for ALS. But by doing that, and by finding out that missplicing is so core to the development of disease, we started to really understand how to develop therapies for splicing disorders, and specifically ones where restoration of RNA and then the subsequent restoration of protein can really have a therapeutic benefit. And so if I look at QurAlis now, we have an expertise in stem cells and human translational models. We have an understanding of genetics of ALS, but also of other diseases like frontotemporal dementia and Fragile X and PSP, where we're also now actively working. And we have technologies that can really develop therapeutics in a pretty scalable manner. We have now a couple of times, both for the StaffMin program and for the UNC13 program, went from a genetic insight to a development candidate in about 2.5 years. And we then know how to get to the clinic in about 1.5 years. And we then know how to run the clinical trials. And so if I take a step back and I look at where we are as a community, genetic understanding of disease, we have technologies that we understand really how to develop drugs in terms of genetic medicines. In our case, really how to restore RNA for therapies for rare diseases. And then we have these translatable models, these patient-derived neuronal models. So I'm actually really excited about that. And then new technologies are starting to emerge. We think we can get to much less frequent dosing. So, we believe that we should be able to get to twice a year dosing. Now, I don't know if our the first therapies will be twice a year, but I think we can definitely work towards that. And then also technologies to cross the blood-brain barrier are starting to get really excited. And so if you combine that all together, I think we're going to see a lot of movements in the next 10 years or so on many different diseases. And I think QurAlis is going to be a big part of that as well. So we are pushing forward as best and as well as we can our ALS therapies. We really want to stop that disease, but we have started to work also even on epilepsy, on frontotemporal dementia, on Fragile X, and on PSP. And as a small company, you need a lot of focus. So that is the challenge together. But in essence, I think we're in a very good time where genetic medicine has matured. Genetic understanding has matured, stem cell technology has matured, and genetic medicine has matured. So I expect we're going to see the benefits from that in the next decade, basically.
I was going to ask you, when you look out for the next 10, 15 years, what is that vision that you have? And, and maybe you answered the question, because I think it's, you've got all these things kind of coming together, but what do you really hope for patients? But then also, what do you hope for your company?
Yeah, I hope that AI actually will have some meaningful contribution here. The big challenge is still the cost of developing therapeutics and the time that it takes to develop therapeutics. Obviously there is a pushback on drug pricing in the society. But when I think about ALS, there's 40 different genes approximately. It depends on where you put the cutoff, all of that. So there's C9orf72, I talked about in the beginning, is 8 to 10% of the patient population. Then the next biggest gene is SOD1. And then you have smaller genes under that, TDP43, FOS, TBK1. If we think about it from a commercial perspective, all these small ones, the long tail, it's very hard to develop therapies for those because it costs too much. And so this is something that we have to solve as a field. We have to make the development timelines shorter and less expensive. And I think a couple of things are really going to play a role there. A regulatory framework that supports it, but also biomarkers that really help us, maybe run smaller trials and shorter trials to get to a meaningful understanding if a drug works or not. Because with genetics and stem cell technologies and genetic medicine, we can move fast. But the traditional drug development path is slow and incredibly expensive. And I know we are moving on it as a community, and the FDA is actually making big steps there as well with surrogate endpoints as for the SOD1 population. But we have to really push that also. If we can do that, then I think our ability to bring meaningful therapies to patients of many different diseases is going to be catalyzed. And that there, I hope QurAlis will be playing a big role in that. My internal mission is always do more, do it better, and do it faster. Because, it's kind of a sentence used a lot, but patients are waiting. I think about myself, if there's family members or friends that are suffering from diseases, you want to help them now, right? I was speaking to an ALS patient yesterday who asked me, what can you do for me now? And I was like, it's hard because our development path is longer. The average life expectancy is 3 years. If you have that type of timeline, it doesn't match up with the drug development timeline. So I think we have a framework, but we haven't solved it all yet. And it's patient heterogeneity, understanding if a drug works, if it's efficacious in a disease that is slowly progressing is incredibly difficult. And so those are not easy hurdles to overcome. We don't want to bring things to the market that don't work, right? We don't want to, have snake oil salesmen, basically. So there's a tension there. But I am very optimistic as a person, and I'm very bullish on genetic medicine. But I also know that there's things we still have to work through to really, bring it to patients better.
Well, we're running up to our time limit here. So I want to give you an opportunity that if others want to kind of reach out, find out what you're doing, maybe collaborate with you, how do people get in contact with you or your company?
Yeah, so we have a really good email address for that, info@QurAlis.com. It's also on our website. If people want to work with us, this is a community. We are part of a community. And so we work together with many different companies and also individuals. And, obviously, if it makes sense for us. But my former chair used to say it takes a village to make a drug. So it takes a village within the company, but it takes a village together with everyone else in the world. And so if there's people that think they can help us move better, faster, and then, please do reach out. We're always looking for exploring opportunities to collaborate.
Well, I am always in awe of people like you. I mean, you're dedicating your life to make things better for all of us, and that passion and that urgency is definitely felt. So, I appreciate you spending the time today and going through kind of the science of it, the business of it, and some of the challenges and the hopes. So thank you very much, Kasper.
Well, thank you for the opportunity, Deborah. And again, we're doing this together. So we're all part of the same mission, I think, to help patients.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.